Okay. Um, yeah, this is work in collaboration with the colleagues in Simula, Ada, Alvor, and Marie. Just to give you a little bit of the context of the talk, it's computational neuroscience. Here you can see a slice of a mouse brain, essentially, and it's very complex tissue. There are many different types of cells, for example, neurons that you may already know, and you also have astrocytes, these star-shaped cells for which the mechanism is not really understood, even if there are many, even more than neurons. And then you have other type of glial cells and myelin sheets. And it's a very complex tissue. And what is interesting is that recently we have very nice data from microscopic imaging and 3D reconstruction, as this one. And here you can see there are different neurons, some astrocytes, blood vessels with some structures wrapping around. And since, for example, the astrocyte is not clear what they do, how they function, it's an hypothesis that they take place like have a role in the wasting clearance of the brain. But there is a lot to know and to investigate in this setting, also because now we have this amazing data. This is an example of the geometry and the meshing we are considering. So of course you have to build the mesh, and this is quite a lot of work considering the complexity and also the detail. So you need a very fine mesh. And also what I show was not really realistic because actually the tissue looks a bit more like this. So it's very packed with cells with very thin layers of extracellular media between them. And it's also quite complex because different cells have different roles, different membrane mechanisms. So modelings can become quite challenging. And with this, we are already able to do some kind of modeling. This, for example, I'm showing here some potential that is triggered by essentially an electric signal through some axons. And there are different cells with different membrane mechanisms, and they react to this signal spreading. What is also interesting is here ephaptic coupling. So it's another hypothesis that is kind of debated. So it's the idea that cells can communicate also between these thin extracellular layers. So this is also something we can study and investigate with simulation in this area. So I show you the geometry. Now, what do we model on these geometries? There are various kind of models depending on the scale and complexity in electrophysiology. You have, once you have like your domain with some extracellular and intracellular space, you can use, for example, the monodomain where you consider just like an electrical potential everywhere. A bidomain, so you have intra and extracellular potential in every point of the domain, or you can go for a very small scale, and this is what we do here. This is the MI, or cell by cell, so we consider the explicit representation, geometric representation of the cells, as I shown in the geometries. And then the KNP model is a model where we consider also concentrations. So the concentration of ions, which are essentially what drives all the signaling in cells. And you have different forces. You have diffusion, and you have like electrical forces, electrical drift, and also the ions both generate and react to these electrical forces. And what is interesting is that you can see here there are different activities in the brain in this experiment from sleep to some disease. And these are the concentration of some ions, potassium and calcium. And this change quite dramatically. So this can be also a trademark of disease and can be interesting to model. So we are using this cell-by-cell framework, also known as MI, extracellular membrane intracellular. And essentially, you subdivide your domain in intra and extracellular, and then you solve Laplacian inside and outside, these two elliptic equations here. Then you define a jump, and you require the solution to be continuous in terms of the gradients along the interface. And then you have some ODE in the interface, where this is where all the complexity and the dynamics comes into play, because otherwise inside and outside is just elliptic problems. And this is just for the potentials. This is an example of the solution for 1 cell or 25 cells. So it's interesting because it's continuous. The gradients are continuous, but the solution is not. But what we're actually doing is this CAN-PMI model. So we also consider concentrations. We have these two equations which model this phenomena. The first one is essentially a continuity equation for the concentration, and you have here for the flux a diffusion term and electric drift term, which depends on the potential, electrical potential. This is your drift velocity. And the second equation is a neutrality equation. So it's a constraint that like essentially your solution is neutral in any point of the space. And then again, you have some condition of the interfaces and some ODE inside that create all the dynamics and the complexity. This is an example of a simulation. So here we trigger some impulses in the neurons that you can see also here. And in this movie, we can see how the electrical impulse essentially triggers gradients in concentration, both inside the cells and in the extracellular media. And inside the cells, of course, you have neurons and astrocytes, and they have active or passive membrane dynamics. So they have to be modeled like differently depending on the type of cells they are. This is another example of just a single astrocyte where we inject some ions here, down here, and then we see how they electrodiffuse in the extracellular through the membrane and inside the astrocyte over time. So a little bit about the mathematical formulation that we use. This is a mixed dimensional problem, meaning that we have both a problem in a volume and a problem at the membrane. And there are two ways to address this problem. One is single dimensional, and the other is multidimensional. In the multidimensional, we keep this multidimensionality also in the system, and we have like unknowns in the bulk, in the volumes, and one unknown in the interface. And so you get this kind of system, which is somehow similar to also you see in domain decomposition with this Lagrange multiplier structure. But what we are using actually is this, where we eliminate somehow the interface unknown, and we end up with this system, which is single dimensional, and it's symmetric positive definite, while this is not positive definite anymore. And for this, AMG-CG works very well. It's robust with respect to all the parameters and also to the number of cells according to some recent studies. And you can look at the spectral properties of the systems and look at the preconditioners. For example, we study how if you uniformly refine, how you can build a preconditioner that essentially removes everything which is on the gamma, on the interface, and you get still convergence and robustness. This is an interesting example for a parameter tau, which is the tau, delta t essentially, going to 0. And you can see that the condition number of the MI system kind of goes to infinity, but the number of CG iterations remains bounded. And this is kind of interesting because classically you would expect this kind of bound, so you're error depending on the condition number of the matrix for the CG. So in this case, the number of CG iteration you're doing. But this somehow contradict this bound, not really because this is an upper bound, so it's not contradicting, but it's a bit counterintuitive. And the thing here is that essentially you have just a few number of eigenvalue going to infinity. And so the condition number is going to infinity, but you have a bounded number of eigenvalues going to infinity. And so in this case, there is theory by access on the stealthy, essentially, that's you still have a bounded number of CG iteration to convergence. The numerics, we use IMX linearization. So the problem that I showed initially was not linear, so we need to linearize. And for this, we do implicit, explicit. We have two different schemes. One is CG plus CG, CG inside and CG outside of order P. And for that, we need something else. This is a multi-Phoenix module, which can deal with the interfaces and a monolithic solver. And this is what I'm going to show. But another option is to use DG, just DG everywhere, because of course you have this jumps. So this G is very convenient with that respect because you don't need any external module. So, but the problem is that you get much more memory. So that's a bit of the trade-off. And for the meshing, we use f-wild, which is very convenient for meshing complex structured surfaces, essentially. And this is the algebraic structure of the CAN-PMI problem. You can see these are the blocks for the concentrations, and they are coupled by the potential block. This is an example of the solvers that we use. Essentially, you can see a direct solver here, and then we employ essentially MG to a block precondition with which decouples everything, all the cells and all the concentration and the potentials. And this works very good. You can see here, for example, for this kind of size here, 1 million degrees of freedom with respect to just plain DG or direct solver or like ELU, you can get down quite a lot with the runtime, staying robust in terms of iteration. And this is also, yeah, robust in space and time. And these are two different options. This is monolithic and this is field split, but a similar message. And finally, this is the scaling plot. So this strong scaling is pretty good. Number of iteration, runtime, it's almost close to ideal. And weak scaling, again, this is the MG GMRS. Is okay, it's not ideal. So if you have some tip to make it this even more flat, this would be welcome. But at least the total runtime is still quite good in yellow. So this is acceptable. That's it. That was my talk. Thank you for attention.